Hello everybody. I'm glad to see you. So we are starting our coffee break with TRANSFAC. So you can grab your coffee, relax, and be ready for to listen about TRANSFAC and its features, and be ready and prepared to put your questions. And today, what we are talking about is basically how to predict, how to find, or give me please target genes for my beloved transcription factor. For instance, we got so many questions like, can you give me all target genes for transcription factor NFkB? That's a good question. But actually, I always ask, in which tissue? You know, transcription factors, they regulate their genes in tissue-specific manner. That's why we have so many different cells in our body, and genes, different genes are expressed in those different cells and different tissues, and that's regulated by different transcription factors, and they're binding to their targets differently in different tissues. So actually, so we know there are many tissues, many cell types in our body, and it's a very nice resource, human protein atlas, where basically all genes in human genes were checked in which tissues they are expressed, and everybody can get this information. We also are getting this information from these resources, as well as from other resources. Actually, we are putting information in our databases about expression of many different genes, including expression of transcription factors. So different transcription factors are expressed differently in our genes, in our different cells, you know. And basically, let me first have a look at one transcription factor and just have a look how does it look like. So this is MEF2A transcription factor, which is expressed in muscle tissues. And actually, it's involved in cardiac, myofibril assembly, and it's also involved in many diseases. There are known associations of this gene with such diseases like coronary artery disease or myocardial infarction. So these kind of bad diseases are connected, related to either mutations or various, malfunctions of these transcription factors. So definitely it is important, and we would like to know where this factor is expressed. And if we get this information from Human Genome Atlas, a protein atlas, basically, we get the profile like that. So where we have all kind of tissues here, and this is expression value of this particular gene in that tissue. And here we see the heart, actually heart muscle tissue, where this gene is expressed maximally, and that's kind of a very specific for this particular tissue. And the higher this peak in relation, in comparison to other tissues, the more specific this gene for that particular tissue. But as you can see, there are other tissues where this gene is expressed. So we can also sort it by type of organs where this gene is expressed, or by expression. So different possibilities are given here. Anyway, so now we know how these genes are expressed, how the transcription factors are expressed. And what we are going to do today is let's predict which transcription factors are actually binding to promoters of my tissue-specific genes. So what we are going to do, we will take the set of genes expressed in heart muscle and analyze it using TRANSFAC database, using the all the motifs in TRANSFAC database to find which transcription factors are actually regulating my genes, which are specifically expressed in muscle tissue. All right, so for that we have a tool. So let's go now to our login. So at our homepage, you can log into all our tools. Here you click login. and you have your username and password, so everything is online. If you don't have your login, you have to register here. So you got your license from your library, for instance, where you put your license key and then you register yourself. And then once you have your login, you log in, sign in, and then you have your all kind of products which you are subscribed in on our site, including these databases of TRANSFAC and other databases like TransPass and Human PSD. But today we are actually really focused on TRANSFAC issues and how to predict tissue-specific transcription factor binding sites. And for that, we have this match tool, match suite tool, which is there to analyze your sets of genes. So as a set of gene, we have I have prepared here a set of muscle-specific, heart muscle specific genes. It's here we have 62 genes, simply a list of accession numbers and symbol IDs. You can also use gene symbol. So that's a list of genes of your interest. For here we have these heart muscle specific genes. So we are going to load this set into this tool, gene set analysis. We click next, and here we basically have to upload my file. So I select it from here, from the examples. So these are 62 heart muscle specific genes. So they are uploading into our system. And then once you have them, we are ready to, so you basically can click on them, see, they are here, 62 genes. So now we click next, and we have to specify what is the range of promoter we are going to analyze for each of those genes. So the standard range is from minus 500 to plus 100 around transcription start site, which is a very standard basic promoter lengths. Of course, you can extend it if you like, so to up to 5 kb upstream and 1 kb downstream from TSS. Here we basically use the standard default, and you have to select your tissue of your interest. So here we are interested in heart muscle. So we have here heart muscle, and then we click next, and we're ready to launch the analysis. So I will explain later the more details which steps are going on right now. So what is happening here? These 62 genes are mapped to the genome, so human genome, the latest build, and then promoters of those genes are automatically extracted from the genome annotation. Then there are preparation of that library. Actually, as you know, we had the previous coffee breaks. TRANSFAC has a lot of motifs. Now it's more than 10,000 different positional weight matrices for different transcription factors of many different organisms. Here in this analysis is focused on human, which means we take vertebrate matrices, so matrices for vertebrate organisms, which are around 7,000 positional weight matrices. And of course, not many of them will find their hits in these promoters. But what this tool is doing, it's trying to estimate the enrichment of those promoters by binding sites for various transcription factors, and it takes into account which transcription factors are actually expressed in that particular tissue. All right, so it's running. It takes a few minutes because it's also doing quite a lot of post-processing of binding sites. It's trying to find clusters of sites, and it's using some machine learning to do that, some genetic algorithms. So it takes a bit of time. But let me basically open ready results. Of course, I was running this set of genes already before. So you click here on view results, and you have here actually these three windows. Let me Let me describe what do you see here. So, okay, so in this right upper window, you see this list of 62 genes. And so they are listed here, and of course they are annotated. And here it's a table with those transcription factor motifs, which are here at the top, and associated transcription factors to those motifs. And again, as you know, then one positional weight matrix, one motif is actually connected to several different transcription factors, because the motif describes this DNA binding domain, and we'll talk about this today. But the whole rest of the transcription factor can be a bit different, and that's actually affects the tissue specificity, and not only tissue, but also specificity to various conditions, how these factors are expressed. But anyway, so we have this table. So for each gene, for each promoter of that gene, we see how many sites were found for each particular transcription factor, which were selected as most important and enriched transcription factor motifs in these sets of 62 genes. How do we which ones were found to be enriched are shown here at the left window, and they are sorted by sort of importance of the enrichment, also taking into account expression of that factor in that particular tissue. So and the top, as you can see, it found MEF2A, so the one which were considered. And actually, if we look at a factor view pro, it shows also which kind of motif it was connected. So it's connected to MEF2B motif. So actually, it's interesting case, for instance, here, that the name of the motif is for MEF2B, but in fact, the binding factor which was found and in association with that motif is MEF2A. Because this particular transcription factor is not only connected to this enriched motif, but it's also very, as we have seen before, it's very specific for that factor, for that tissue. So for heart muscle tissue, it has a maximum level expression. And also here it shows that if you rank all other tissues, all other 62 tissues where this factor is expressed, then the heart muscle tissue will be the number one. So it's not only highly expressed in that tissue, but it's actually maximally expressed in that tissue. So it's very specific for that tissue. So the second factor is SRF, which was found by SRF motifs. And actually the motifs themselves, you can see when you click here of matrix view. So you see the motifs which were found enriched. And here you see this enrichment factor. For instance, the top enriched motif is actually SRF. And the enrichment factor is adjusted enrichment factor is 4.8. And actual enrichment, which was measured even higher, like 1159. So which means that it's how many times number of found sites in the promoters of those genes are higher than in random promoters. So if you pick up random promoters in the human genome and compare the frequency of those binding sites for SRF in the random promoter to the frequency we found here in the muscle, heart muscle specific promoters, we see that 11 times frequency is higher here. And when we adjusted this frequency towards, statistical adjustment, we made statistical very, very stringent adjustment. Still, we see enrichment around 5 times. Also, there are a number of p-values which are calculated for that, so telling whether the number of sites found in these promoters is much higher than expected by random chance, and p-value is very significant. And there is a sequence enrichment which tells you amount of promoters in my set of 62 genes where at least one binding site for this factor was found is also much more than it is expected by random chance. So which is also estimated by p-value. So in all respects, this motif is very important for regulating those genes. But from a point of view of factor, indeed, SRF factor connected to that motif is in this list. But even more interesting is MEF2A, which by enrichment shows maybe not as high as SRF. So you see it's like it's only 3.8, but still quite high. But from point of the specificity of expression, this factor is very important. So you see, we listing these factors from both criteria, from the criteria of enrichment of the motif, and from criteria of expression of this factor in that particular tissue. All right. So, and then of course you see here in each promoter. And to see that in details, we have this window here, which basically shows you exactly the promoter we are focusing now. So we clicked on the very first promoter, myosin light chain 3 gene. And you see here in genome browser, you can actually zoom out a bit more or zoom in. You see this is a gene. And in fact, that's a very interesting case because this gene has introns and exons in the standard annotation of genome, but promoter which is acting in heart muscle is located in the first intron of that gene. So that's the point, that we don't only consider here the expression of the transcription factors, which are tissue specific, but we also consider the promoter of that gene, which can be also tissue specific. In one promoter, in one tissue, the promoter of that gene could be here, in another tissue can be there. So we have this information in our database, and we take this into account. Okay, so there are some questions, more technical questions. I will answer later. Okay, so we can zoom in a bit more to look a bit more in details here. You see what's happening here. We have here each arrow here is one predicted binding site for a transcription factor. For instance, the ones we were talking about, this SRF site. And when you click on that site, you see the consensus of that motif. You see also what is the score of that site in that particular gene, in that particular position, and what is other criteria. And also you can click here on the accession number of that on that positional weight matrix and go directly to the TRANSFAC database to get more information about that matrix, how it is, what was generated. So this particular matrix actually generated by Selex experiments, and actually there are other matrices available in TRANSFAC for that family of factors. You see they are slightly different, but the consensus is mainly the same. And in this case, you see there are many matrices for these factors, and they were all checked in that analysis, but as you can see, only one gave the significant enrichment, and that's also the value of so-called redundancy of many different matrices for the same factor in our database, because in different conditions, in different tissues, the factors can function in various complexes with other cofactors, which are different in different conditions, and that may influence on their binding specificity and binding pattern. That's why we're collecting all kind of positional weight matrices in TRANSFAC from the various literature, from various databases, and keep them all, and then let the algorithm actually find the true one, the one which is better fit to the conditions we are analyzing here. Also here you can actually put more information on that browser. For instance, you can put information about experimentally validated transcription factor binding sites, information about conservative regions in genome, which are also quite important. And from that point of view, you see we actually have this SRF SRF binding site known, experimentally known for this gene, and which fits very well with our prediction. So actually, if we click on this site, we also can go to TRANSFAC database, but now to a different part of it, which is describing each individual sites which were experimentally found. And this particular site was discovered actually many years ago in 1990. It was the time when many people were interested to study each individual sites, and they did a lot of experiments in human and also in mouse in the orthologous gene promoter. And they actually validated this binding site in that particular conditions. And as you can see in human, it was found in HeLa cells. It was not in actually muscle cells, although in mouse it was also found in skeletal muscle cells. So in that respect, this factor which we see here, this binding site is fitting well to the conditions we are studying. Although maybe in other cases, we may find sites which are experimentally proven, but they are proven in completely different conditions, which are not well relevant for the conditions we are studying right now. So that's why predicting binding sites using such tools is of tremendous importance, because we are not only finding potential of binding, but we are really focusing on particular conditions where these factors are binding to those promoters. And one more thing is about the conservative regions. And you see these regions are conserved, and that's exactly fitting to the region which was corresponding to this SRF site, as well as some other adjacent region. And as you know, if such region is conserved, it give additional evidence that it might be functionally important, which is of course true in many cases, but not exclusively true. So there are many transcription factor binding sites which are very specific for human, but maybe in mouse this region is absent, but still they are regulating their genes. So this criteria of being conservative is important to find few marker binding sites for transcription factors, but it's not exclusive. So actually, you have to consider also other binding sites for even if they are not located not in fully conserved regions. All right, so we have here the first look at these results. Going a bit more in detail of the results, we can actually click on report, which is generated automatically by this tool, and it's gives you a summary and also all those tables which we showed here. Also maps of the binding sites and promoter of the genes, where you can see the top ones, and if you click here, you see the full table of all the maps. Okay, so there are also technical considerations anyway. So right, so and actually here there is one interesting point. As I mentioned in the beginning, that one thing which is this tool is doing, it's doing a lot of pre-processing of the binding sites which it found. So first of all, it looks for combinations of binding sites, and that's very important for tissue-specific expression and tissue-specific regulation, because those factors, they, as I said, they never regulating their genes alone. They're regulating them in complexes. And here we have a tool which is called CMA, Composite Module Analyst. And later, in further coffee breaks, we'll be spending quite a bit of time on understanding how this tool works. But basically, what it's doing, it's trying to build a model, trying to build a complex of few factors, few motifs of the transcription factors. And here you see it found like 2 modules. We will see what module means. which include SRF with a few other zinc finger factors, and RF factor with a few other factors around it. And when this complex is considered, then the promoters of these muscle-specific genes, heart muscle-specific genes, are clearly differentiated. You see these red bars from promoters of other genes. And it's usually, basically this tool is trying to find such combination of factors which discriminate these promoters from other promoters. And by doing that, it's not only finding the most important factors, but it also found the combination of factor for actually working in one particular place in the promoters of those genes. We will see that a bit more when we look at each promoter. All right. So if this part with how to run the tool is clear to you, so I will go a bit further with understanding how these transcription factors look like. So you have chance now to give you more questions if you like. Just type them in Q&A. If you don't understand anything, just drop a line saying, "Okay, can you explain a bit more one of the steps before?" So I will have time to return to that and to make it more clear. But okay, so let's then go further and understand a bit more about this tissue-specific regulation. So as you know, gene regulation is actually governed by not only by promoter region, but by many enhancers which are located in a bit distant regions, not only in the region near start of transcription, but it can be extended in introns, like we've seen in case of tissue-specific promoters. The whole promoter is located in intron, but it could be the whole specific enhancers which are regulating by genes. And the regulation of that in one particular tissue is actually governed by many different influences from enhancers from the genome. And if you look a bit more in details how those enhancer regions look like, they actually look very similar in a sense to promoter regions. So they have a bunch of binding sites for various transcription factors. So it's like hierarchical organization. So you have the whole 5' region, including all promoter and enhancer regions, which can be split into subregions, which we traditionally call an enhancer. But in fact, that's a bunch of binding sites for various transcription factors, which are colocalized, and they together, by combining to each other and by acting on the promoter region, on the start of transcription, they initiate or regulate the transcription. So, and as I showed you before, we have those matrices which are constructed in TRANSFAC by known binding sites. So that's all you know. And these matrices are used to predict new binding sites for transcription factors. That's what we are doing here. But then let's have a look at various transcription factors. So the transcription factors are split into families, and more upper hierarchical levels. So for instance, if we look at the transcription factors IRF from a family of interferon regulatory factors, you can see here that there are like a group of them starting from IRF1, IRF2, and up to IRF9. And these are classification for human. And in other species, we have even more of them. And then each of that factor is characterized by a particular motif in DNA where it is bind. But you can see these motifs are very similar, and that's the reason is because all of these factors, if you look at them, they have the DNA binding domain, which is very similar to each other. In some cases, it's actually identical, but at least it's really, really similar. And that's why the binding where it is, which is done by this DNA binding domain, is very similar, and which means motif where these factors are binding basically the same. But each of that factor has a different other part of the protein. So-called activation domains, which are shown here, and all other kind of domains, which are regulating how these factors are actually working. So DNA binding domain is just to anchor the factor to the DNA and how it is working, in which conditions, which other factors it's interacting. It's all done by this other type of domain. That's why in different tissues, by the way, here you can see alignment of these factors, and as you see, this gray part is alignment of the DNA binding domain. As you can see, very similar alignment, but the activation domains, well, other part of the protein is very different. So basically, you cannot align it to each other. And now, if you look more in detail for where these factors are expressed, you can see the different pictures, different profiles. For instance, IRF1, IRF2, and IRF3. Well, IRF1 and IRF2, more or less, they have very similar pattern of the expression. And this blue part is actually immune cells, various types of immune cells. Whereas, and also, for instance, IRF7 and IRF8 are quite specific for immune cells. But the factor, for instance, IRF4 is expressed in other types of tissue maximally, and actually IRF6 is completely different. So it's expressed in other tissues. I don't see the name of the tissues. We can zoom it in, but you see the profile is completely different. And IRF3, in fact, is very much ubiquitous. It's expressed in many different tissues, and which means all these factors have very similar or more or less identical DNA binding domain, and but the factors itself, they have a very specific action in different tissues, which means if you analyze now different types of sets of genes coming from different tissues or different cell types, you may expect different transcription factors to be important for regulating my genes. For instance, you can see here, we can actually zoom in. Maybe I will do it like this. All right. So if you look at IRF1, so it is expressed in top expression in granulocytes, right? And for comparison, the RF8 is expressed in dendritic cells. Both of them are immune cells, but they have a very different function. So I have here pictures of dendritic cells and neutrophils. Well, granulocytes and neutrophils are actually one subtype of granulocytes, and they have different functions. For instance, dendritic cells are actually important for immune response when it's presenting antigens. But granulocytes are more important for innate immune response. So they have particular different function. Although IRF factors are working in both cases, but different subtypes of that. And to illustrate that, I run actually match suite again for these two types of cell types, granulocyte and dendritic cells. And let me show you how I did that. Basically, we have one special tool here in the match suite, which helps you to find genes for a particular tissue or particular cell type. So here is called construct tissue-specific gene list from scratch. So if you don't have your gene list, you can construct it directly in this tool. So you click here, you click next, and then you select your tissue. For instance, we select here dendritic cells. Then it will show such a graph to you, which have two axes. And here, each dot is one gene. And here is expression of that gene. So which means in that particular tissue or cell type in dendritic cells. So this gene, for instance, has a maximum expression, right? And this axis is a tissue specificity. So you know that actually it's a very interesting measure. We measure it from 0 to 1. 1 is absolutely specific, which means this gene would be not expressing at all in other tissues and have just one peak in that particular tissue. It will be specificity 1. For instance, this gene has specificity 0.8, although the expression itself may be not very high. So it's a kind of trade-off. Either you expressed everywhere, more or less, and not very specific, but you expressed a lot. But here we see 427 genes, which are cutoff of tissue specificity in dendritic cells. You can select, if you like, you can select all of them in your subset, like 427 gene, or you may select only very specific ones, like 38 genes, and proceed with that set of genes. So basically what I did, I selected all of them and proceed with selected genes. So it tells me which genes these are. And then I can click here on the green box and proceed further with this set of genes. So now I have these 427 dendritic specific genes, and it's already put me here, the dendritic cells, and the promoter region is ready here. One actually important thing which we can do, and it's interesting to do, is to see these 427 genes. Although they are specific for the dendritic cell, but they are doing different things. And they may be actually, by doing different things, they may be be regulated by different transcription factors. So you mean, you see that transcription factors in their combination not only specify the tissue specificity or cell type specificity, but they may be different for different functional categories of the genes they are regulating. So, and what it's doing now, it took these 427 genes, and what we are doing, we're mapping them to gene ontology to see what type of gene ontology functions these 427 genes are doing. And you see, for instance, here it's a big set of GO terms which belong to oxidative phosphorylation. Genes which are in the dendritic cells belong to that particular GO category. But for us now, it's most important is immune response. We can double click here, and we get these 160 genes, which are not only dendritic specific, but also they are known in GO category of immune response. So, and then we proceed with further with these 130 genes, and then we launch the analysis as well. So let me show you the results of that analysis. Actually, I will stop this one because maybe it will slow down the server. So I want to show you the difference. So I have these dendritic cells analyzed, and also granulocyte specific genes. So in both cases was around 420 or 416 genes which are specific for these different cell types. And again, I ran them through the gene ontology and selected a subgroup which belong to immune system. And now I can view the result. These are dendritic cells, and as you can see, RF factors, which are known to be responsible for regulating genes in immune response. And here, indeed, RF factors. And from matrix view, you can see this RF must be here, maybe not at the top, but from factor view. So this RF8 is at the top of our prediction. So we indeed found it connected to IRF4 matrix. So actually interesting that this matrix is created was created from IRF4 factors by a ChIP-seq experiment. So actually, at some point, these people, this was compiled from the Hokomoka database. So they did ChIP-seq experiment, and from that ChIP-seq experiment of IRF4 factor, we build this matrix. But this matrix is used to predict IRF factors in general, because as I said, the DNA binding domains of those factors are very similar. And at the end, IRF8 in this case is predicted to be important for this particular cell type. And if we now look at the other analysis which was done for granulocytes, so we can have a look here. And again, we see here IRF factor at the top, but now it's IRF1. So the motif itself is again the same, IRF4, which was it's a very good motif, it seems, for predicting various types of IRF factors. But since we know which cell type we are analyzing, we can predict particular factor which is actually regulating my genes. All right. I want to show you also one additional thing in the report. I guess it's in the report of dendritic cell. Let me look at this report again. Here, we analyzed. Let me have a look at the report. And you see, by the way, here these GO terms which were used here. And what I want to show you is this heat map. Okay, let me zoom in a bit, even more. What it is? Actually, it's a bit complicated, but quite useful. So what you see here is a heat map of GO terms. So we had this 400-something genes. No, here is a subset of genes, 160 genes. And then these 160 genes were further classified by GO terms. And then we compared which GO terms are similar to each other by these genes. And then we see here, we can split the GO terms into subcategories. So for instance, here, like immune system process, immune response, cell activation, this is like one block of GO terms from the perspective of these genes. And you see here the top of log-fold, actually p-value of belonging of that set of genes to that particular category is very high. So which means this each subcategory of GO terms here is quite significant. But what is interesting here in front of these GO terms, we see another heat map of various transcription factor motifs. And they have this color, which tells you in which particular subcategory this motif is the most significant. Like you see here, this IRF4 motif, which we found to be significant for the whole set of 160 genes. But actually, if you look in more details, like we do here, you see this red box here. This says one is actually corresponding to cell activation. And another box here is leukocyte-mediated immunity, cell activation evolved in immune response. So a very particular subcategory of GO terms, which is particularly enriched by binding sites for this factor. So by doing that, you actually can dissect your set of genes into smaller subset and understand which of the functional category of those genes are regulated by which transcription factor. All right, so that's like a bigger picture on our analysis results. At the end, what I want to show you also is actually a bit more in depth of the files which we generated. So actually, this view is overview of the results. but actually the real results, files which are generated by multiple algorithms here are collected behind the scene, and you can actually look at them by switching to here the right upper corner to the platform. And the platform is a set of tools. It's a platform. So actually, if you click here, the analysis, subpart of that platform, you see a lot of tools for site analysis, for functional classification like geoterms, pathways. You see a lot of tools. Also, there's a pathway analysis, by the way, which is not touched today. And the data which we analyzed are located here in our project. And if you look at that, results, so for instance, in this granulocyte dendritic cell analysis, see here you see the report which we were looking, but here you also see all the type of results all the subtypes, all the subfiles, and then basically you can look at each of them. So for instance, if we are interested to see more details of those composite modules which I was talking about, those compositions of binding sites, so it is located here. And here you can see those modules in each individual gene, in each individual promoter. Actually, if we sort it by the score, so we see those genes, the first exon of that gene, or somewhere the intron of that gene. In cases like I explained that in a particular tissue, this promoter can be not necessarily in front of the first exon, but in most cases it is. So, and then this region of 500 nucleotides upstream of that first exon. And here you see this cluster of sites found by this genetic algorithm, which are found to be composing the composite module. So you can have a look at each of that site in more details and see their scores and the consensus of that site and so on. So you also see basically the histogram of discrimination. Well, in this case, it's maybe not as good as for muscle-specific genes. This set of genes is more noisy, but still it's quite significant, discriminating promoters of dendritic-specific genes from other random promoters. You can also look at them on each individual sites on genome scale, and here we have a specific possibility to show it in genome browser. So I would like to let me actually do it in the other way around. So we can have genome browser with the genes, and then we can put here the sites found in particular gene promoter, and then you see it's really in the genome browser scale. So you see this gene and the promoter and individual sites inside this gene. And as I said, by clicking on each site, you see more information about it. All right. So I think with that, I would like to finish for today with my demo. We actually have plenty of time to answer your questions. All right. So I have here a question, the following. In my research area, plant pathogenic transcription factors are not enough available in Jaspar. Can I proceed with such species when experimentally validated TFs are not available in the database? All right, so this question was about the Jaspar database, so and other species than human. Indeed, TRANSFAC is somewhat kind of interesting in the sense that we collected quite a bit of motifs from different species, not only from humans. For instance, we have motifs from plants, from other model organisms. So let me share again my screen. I can show you that. Right. So here. So if we go to the platform, here among the databases, we have databases from different model organisms, as you can see from human, of course, not only the latest build, but some other previous builds, but also mouse, rat, nematode, yeast, zebrafish, and that's mainly it. Those which are prepared in the platform and ready for analysis using various pipelines. But actually, any new species can be uploaded into the platform, and then can be analyzed. Maybe not with those ready pipelines, but with individual tools, which are here in the analysis section. And I'm planning to have the whole coffee break related to those different model organisms. For instance, here I'm checking if I was analyzing once a genome from special genome. It's here. No, it's not here. Okay, so I uploaded special genome here for some coffee break. I think it was from bovine, and then I showed how to analyze it. So you can analyze different genomes, and also you can use a library of positional weight matrices from different type of organisms. In TRANSFAC, for instance, we have matrices. You can look at them at profile at TRANSFAC. You see there are matrices from vertebrates, but there are matrices for invertebrates, for insects, for fungi. Well, there are very little for bacteria, though, so it's not specifically big collection for bacteria. But others, like fungi, for instance, is a very good collection of positional weight matrices, which you probably cannot find in other types of databases. All right, so I have another another question. We find an unknown zinc finger protein. Can I use your tool to predict the target genes? Wow, that's a very good question. Thank you very much. So it's a zinc finger protein, exactly. So, and for zinc fingers, there is a way to actually assume, not to say to predict, but to assume or to calculate what could be a binding pattern for that factor. We don't have such tools, but I know actually there are approaches, and they were developed in bioinformatics for many years, specifically for zinc fingers, for zinc finger proteins, that just from the structure of zinc finger protein, you can actually make a certain assumption of the positional weight matrix, which can be used for this zinc finger protein. Well, we don't have such tools, but what other thing you can do is if you have your protein, I guess you can generate antibody for this protein, and you can make a ChIP-seq experiment with this protein if it's indeed DNA binding. And then for the ChIP-seq experiment, you can upload it in our platform, and we have a number of tools which can predict the DNA binding motifs de novo. Actually, there are tools which one very good tool is called ChIP-monk, and there is another tool for that which is actually also quite well known. It's called Homer. So those tools we have in our in our platform, and you can use them to predict the de novo motifs for this zinc finger. And once you predicted it, this motif immediately is created in the form of a library which is available and enabled for the match analysis together with other matrices. So actually, also it can be your new motif can be used by this tool together with existing motifs, and you may in principle even find some composite element, composite module, which consists of your motif for your unknown factor together with other motifs for other known factors. So this all possible, but again, it depends on your data. If you have just ChIP-seq data, you are welcome to try it here with our tools. All right, so I have another question here. Any cross-reference database for transcription factors? All right. So if you are really talking about transcription factors, so like proteins, indeed, the very good reference database is a SwissProt. It's a description of all proteins in general, but and certainly transcription factors are also there. But actually, very important thing, and we didn't talk about this today, that transcription factors are complex proteins. So in many cases, if not in most of the cases, they are organized in dimers, like heteromers, with homologous, peptides, or even bigger constructs. And then this whole construct is called a transcription factor. For instance, transcription factor AP1 is actually a heterodimer of two proteins of C-phos and C-Jun, and actually many different combinations of the C-phos, phos and Jun proteins can organize different types of AP1 factors. So when you're talking about a database of transcription factors, you have to take into account these combinatorics of heterodimers and all kind of things. In TRANSFAC we are trying to collect as much as possible this kind of information. What other databases, reference databases about these complexes? I don't know, except as I said, SwissProt, but actually SwissProt is focused on single proteins rather than on complexes and heterodimers or homodimers. Okay, another question. Question we have just received via info email. All right, that's already retyped. If we are analyzing a transcription factor gene, and the question is to find the downstream genes of this transcription factor in genome of that organism, how the downstream genes can be identified? Exactly. All right, so the question is, so basically you are analyzing a particular transcription factor gene. Like in my example, sorry, I showed to you this gene MEF2A, which encodes the transcription factor MEF2A. And now you want to see which genes are actually genes are controlled, expression of which genes in genome are controlled by this transcription factor, MEF2A. And in my example, I started my analysis not from whole set of genes from genome, but a particular subset of those genes, only those genes which are expressed in the tissue of my interest. In your question, you are interested in all genes in genome, right? And I would say this question has no answer, no direct answer, because as I said, this transcription factor may regulate this gene in one tissue, but not regulate this gene in another tissue. Or maybe in that sense, more or less all genes in genome can be regulated by one particular transcription factor, depending on which condition you are interested. So it's not enough to ask, what are my target genes? You have to ask, what are my target genes in a particular cell type, in a particular tissue, in particular situation, under particular stimulation. Saying that, still, if you would like to find all potential binding sites for a particular transcription factor in all genes in genome, that's also possible. And that's done then in our platform in using the tools here. And here we have a tool which is called site search on gene set. And this site in this case, this gene set could be just one gene, right? But wait, other way around. So you want to find all gene. You want to search in all genes, but for one transcription factor. Very good. So basically, you have to put here the set of all genes, all 35, or 30 or 50,000 genes in genome. We have some such subsets of genes in, I think, in a data set here. Like we have housekeeping genes, and we have promoters, all promoters of the genes. I don't see here all genes of genome, but this can be easily generated, the list of all genes in genome. So you put it here, and then you need to describe here the profile, which is a set of positional weight matrices from one transcription factor. And that's can be done here in our database. So basically taking all matrices from, for instance, you are interested in vertebrate matrices. So you take all the matrices from human, and then you select only those subset of matrices which are connected to one particular factor. So there's several steps of doing that, and it's a good question. I bet I will show it in one of the next coffee breaks, because it has like four or five steps we have to do. You have to find this set of matrices, you have to convert them, and then you are ready to do this analysis. And once you do it, you see all sites in all genes in genome for your particular transcription factor. Thank you for your question. All right, so we have another question. If we are analyzing a transcription factor gene, and the question is, okay, that's the same question. It was just duplicated. All right, so that was the question about finding a transcription factor, one transcription factor, and all genes in genome. I'm promising I will tell about this in very many details in my next webinar. But I promise it's possible here in this tool. All right, thank you very much for your attention, and come next time. See you.